Hello everyone, I'm Dr. Malik Siddiqui and welcome you all to my YouTube channel. And in the series of my lectures, Understanding Immunology, today I'm going to start my lecture number 2. And my lecture number 2 is on the cells of the immune system. Before discussing the topic, let's have a recap of our previous lecture that I gave you on immunity. Now, immunity can be of two types: innate immunity, which has got feature of rapidity, that it is a rapid response, quick response, and therefore it is a generalized kind of immunity which will halt the infection, but it has got no memory. On the other hand, the second type is adaptive or acquired immunity, although it is slower or delayed response, but it clears the infection and it has got a memory for the future also. Now, the overview of the immune system. The immune system uses certain cells, organs, circulatory system like blood, lymphatic system like lymph. So, coming to the cells, which will be our today's topic, in the innate immune response, several cell types are involved, and in the adaptive or specific response, we have got lymphocytes which are involved. Then the organs of the immune system, which will be probably my next topic. Organs can be divided in two types: primary and secondary. Primary organs are those where the lymphocytes they develop and mature, whereas the secondary organs are those where the mature lymphocytes and antigen presenting cells interact to initiate a specific immune response. Now, the white blood corpuscles or leukocytes, there are two types based on the presence of granules. So, if granules are present, they are called as granulocytes, like neutrophils, snuffles, and basophils. And if granules are absent, then they are called as non-granulocytes, monocytes, that includes the monocytes and lymphocytes. So, our today's lecture will be on these cells. Now, as you know the difference between the plasma and the serum, the plasma is the clear-cut portion when we are using the anticoagulant. Okay, and if we are not using anticoagulant after the coagulation, then the clear-cut portion which you will get is the serum. So, after centrifugation in fecal, leukocytes are found in the Buffy coat. That is the 1%, and the other part will contain the RBCs. Now, the cells of the immune system arise from pluripotent hematopoietic stem cells through two main lines of differentiation. So, the differentiation is because of two lineages. One is called as myeloid lineage, which produces phagocytes like neutrophils and the other cells. Now, the lymphocytes are produced by the lymphoid lineage. Now, pluripotent hematopoietic stem cells, they give rise to second generation stem cells with restricted lineage potential, and these are called as progenitors. In this slide, I have indicated 4 points A, B, C, D. A represents the hemosiderin, which is a protein that stores iron in the body, and this iron is derived mainly from the hemoglobin, which is released during the hemolysis. Now, the spot B represents the erythroid precursor from where erythrocytes will be generated. The band cells is spotted as C, which comprise of neutrophils, and the D part which I have shown in the picture are the megakaryocytes, which will give rise to platelets. So, this is hematopoiesis. Now, in this picture, I have shown hematopoietic stem cell which is capable of self-renewing. It can give rise to two kinds of progenitors: myeloid progenitor or lymphoid progenitor. Now, the myeloid progenitor, it can give rise to either dendritic cell or it can give rise to other kind of progenitors. Like for example, number one is the granulocyte monocyte progenitor, which give rise to monocytes and macrophages and neutrophils. Then we have got snuffle progenitor, which give rise to snuffles. The basophils, they are derived from basophil progenitor, and the megakaryocyte, it give rise to platelets. Now in the last we have got erythroid progenitor, which give rise to erythrocytes. Now the lymphoid progenitor can give rise to different types of lymphocytes. Number one is the natural killer cell, which is shown as NK cells, or it can give rise to dendritic cell. And then we have got T cell progenitor and a B cell progenitor. B cell progenitor will give rise to B cell, which will then give rise to plasma cell, and that will produce antibodies. Now the T cell progenitor can give rise to two types of T lymphocytes. One is called as T helper cell and the other one is called as the cytotoxic T cell. Now the granulocytes, they are the first or front line of attack during the immune response. So they are the part of the innate immune response. The granulocytes are identified by characteristic staining patterns of their granules. And the granulocytes are released in contact with pathogens. They produce certain proteins which have got distinct functions like killing, regulation of other cells and tissue remodeling. They all have a property that their nuclei is multi-lobed, various lobes. So number one is the neutrophils. Neutrophils are one of the main effector cells in the innate immune system. Neutrophils comprise of about 50 to 70% of the total white blood corpuscles. They are released from the bone marrow, then they will circulate in the blood for 7 to 10 hours, and then they will enter the tissues, and there they will live only for a few days. Now the number and recruitment increases during the infections. That is, their number increased during the infection. So overall, the number of leukocytes is increased. That is called as leukocytosis, and it is used as a diagnostic feature for any kind of infection. Now the neutrophils are shown to kill microorganisms by phagocytosis, and it was around 100 years ago. The neutrophils are also the main cellular component of pus. The neutrophil name based on staining qualities of the granules. Their nucleus is multilobe. Therefore, they are also known as polymorphonuclear leukocyte (PMN). The neutrophilic granules stain lightly blue to pink. They remain in the blood for 7 to 10 hours, then migrate into tissues. Neutrophils are the first responders, and that is because of their motility and their phagocytic nature. Now, leukocytosis, that is increased in the number of leukocytes, indicates infection. The neutrophils, they are kind of extracellular traps. This is a picture of the slide in which I am going to show neutrophils. Now you can see the multilobe nucleus is present. The second type are the basophils, which comprise less than 1% of all leukocytes. These basophils are non-phagocytic in nature. The nucleus obscured by coarse blue granules, and the basophils are important in some allergic responses, and they are critical to response to parasites. These basophils, they bind the circulating antibodies and they release histamine, and histamine then increases the permeability of the blood vessels. This is a picture for basophils. Now you can see the highly resolved picture of the same slide. Now you can see the multilobe nucleus. Now come to the third type, that is the snuffles. Snuffle has got a bilobe nucleus. Snuffles are bilobe nuclei. They are motile in nature and they are phagocytic in nature also. They are involved in the killing of antibody coated parasites. So, if there is an infection of parasite, the antibodies will bind to that parasite, and then eosinophils will be involved in the killing of that antibody coated parasites. And this is achieved through the degranulation of substances, and which will be responsible for killing the parasites and the worms. This is a picture for eosinophils. Now you can see the highly resolved. Now you can see the two lobes of the nucleus. So this is the picture of eosinophils. Now come to monocytes, macrophages and dendritic cells, which are together called as myeloid antigen presenting cells. They all are phagocytic in nature. They are capable of ingesting and digesting into peptides, and they are present on the cell surface. These cells, they are the bridge between innate and adaptive immune response. These cells, they make contact with the antigens in periphery and then interact with lymphocytes in the lymph node. They secrete proteins that attract and activate other immune cells also. Now number one is monocytes. It is mononuclear, single nucleus. They circulate in the blood for about 8 hours. The characteristic feature of monocytes is the bean-shaped nucleus. I will show the picture. This is the picture, the slide showing the monocytes. So now you can see the bean-shaped nucleus. Now the highly resolved, you can now clearly see the nucleus is bean-shaped. Now the macrophages, as you know, as we all know that, they are will involve in phagocytosis. That is, the first internalize the whole organism, and then it will digest, it will kill the whole microorganisms. The different parts are also shown, like phagosomes, pseudopodia, phagosome, again phagosome, and then lysosome and phagolysosome. So the lysosomes, they contain the digestive enzymes of various kinds which are involved in the digesting the whole microorganism. This is the slide. This is the slide of macrophages. Now comes to the dendritic cells. Dendritic cells are heterogeneous in origin. That is, they can be derived from either myeloid lineage or from lymphoid lineage. They are the best antigen presenting cells for presenting to naive T cells. Ralph Steinman discovered them in mid 1970s, and he just received Nobel Prize in 2011. The name for long processes actively extend and attack sampling antigens and examining the T cells. So dendritic cells are named for long processes, which is got, which are actively extend and retract sampling antigens and examining T cells. The dendritic cells, they capture antigen in one place, then migrate, and then they present antigen in another place. For example, in the lymph node. So therefore, they are called as antigen presenting cells, and they are the best antigen presenting cells. From immature to mature, they change in functionality from antigen capture to antigen presentation. This is the picture of dendritic cell, and this is the slide in which you can see clearly the dendritic cells. Now comes to the lymphocytes, which comprise 20 to 40% of the white blood corpuscles, so leukocytes. These lymphocytes cannot be distinguished morphologically. There are three types of lymphocytes: T lymphocytes, B lymphocytes and natural killer cells. Now T lymphocytes are further classified as T helper and cytotoxic T cells, or also called CD4 and CD8 cell. CD4 cells are involved in recognizing antigen in context to MHC2, that is the major histocompatibility complex 2, and the cytotoxic T cells, they recognize antibodies in MHC1, that is the major histocompatibility complex in number 1. These are the topics which I will cover in my later on lectures, so don't worry about these terminologies. Then we have got B cells. The B cells give rise to plasma cells, and then finally the plasma cells, they will produce antibodies. And the natural killer cells, they are the part of the innate immune response. T and B lymphocytes, they have got large nucleus with dense heterochromatin. The feature of these lymphocytes is that they have got a thin rim of cytoplasm. They recognize specific antigenic determinants. What is antigenic determinant? These are the sites on antigen where the antibodies bind. And therefore, they are responsible for the specificity and the memory of the adaptive immune response. This is a slide in which I am showing the lymphocytes. Now, the resolved picture, it shows the thin rim of cytoplasm. Now, the examples of lymphocytes which I have shown by picture. Now you can see the lymphocyte with thin rim of cytoplasm. The lymphocytes can be of T lymphocytes and B lymphocytes. Then in T lymphocyte we have got helper cell. Then we have got cytotoxic T cell, and we have got B cells. B cells are responsible for production of antibodies. Now you can see the picture number C is a plasma cell which will give rise to antibodies, which will produce antibodies. Then we have got third type of lymphocyte, that is called as the natural killer cell, which is the part of innate immunity. So this is the end of today's lecture. Thank you very much for listening. Please like, share and subscribe to my channel, and I am waiting for your valuable comments. So once again, thank you very much.